Hi, my name is Natasha Cook. Hi, everyone. My name is Natasha Cook. I'm currently a junior at Pitt on the pre-med track, which I've told a lot of you already in our discussion posts, but I'm excited to walk through one of the most fascinating and underrated sensory systems that we have, which is chemoreception. So as you can see from the title slide, chemoreception, from molecules to reception. As you can see, chemoreception begins with molecules in our environment and ends with how we perceive them as smells or taste. Now, in this presentation, I'll explain how the senses of smell and taste work, how they communicate with our brain, and how they shape our behavior and our emotions. I'll also share my own personal experiences losing these senses during the COVID-19 pandemic back in 2019 2020. Which gave me a deeper appreciation for how essential they are to everyday life. I don't think I really grasped how necessary these are for our daily living. Now, chemoreception is known as how we detect chemical stimuli through smell and taste. These senses are ancient, obviously, but they're critical for survival. A fun fact, just that I know based off one of my old classes, is that our sense of smell was used, our sense of smell and our sense of taste was used to detect poison back when, we weren't as modernized as we are today. But anyway, this whole slide just kind of is an overview of what we're going to be going through. Olfaction and gustation are the two senses that we'll be talking about. Olfaction is our sense of smell, gustation is our sense of taste, and they both begin with specialized receptor cells that detect molecules and end with perception in the brain. Now, together they tell us what we're eating, what's safe, and even have the ability to trigger memories and emotions. These chemical signals are converted into action potentials and then interpreted by the brain to form our experience of flavor, danger, or comfort. It's similar to most kind of like neurological processes that we have in terms of getting things through our body to our brain. Now, to go into a little bit more specifics about each sense, olfaction, again, our sense of smell, begins in the olfactory epithelium in the nasal cavity. This image, which I pulled directly from our textbook, shows how olfactory receptor neurons, or ORNs, extend cilia, which are hair-like projections, small hair-like projections. The olfactory receptor neurons extend the cilia into the mucus layer, where they are able to bind to odorant molecules. Now, once an odor molecule binds, the neuron sends a signal through the first cranial nerve to the olfactory bulb, and from that point, information is able to travel through the olfactory tract to the olfactory cortex in the limbic system, which bypasses the thalamus, unlike any other senses. So, the main thing about the olfactory system is that it has no thalamic relay. That's a big thing, especially for neurologists, because it was such a conundrum at the time. But the diagram, it highlights each part of this journey. It starts from the nasal cavity, where molecules dissolve in the mucus, to the synapse in the olfactory bulb, and finally into the areas of the brain which are involved in memory and emotions, like the limbic system. You can see how tightly packed and organized the olfactory neurons are in this image, and how they converge onto specific regions of the olfactory bulb, which allows precise odor discrimination. Now, this direct and structured pathway explains why smells are so emotionally powerful. A scent can almost instantly trigger a strong memory or feeling. I know somebody has you smell something, you're like, oh, that reminds me of whatever the case may be. You know what I mean? That's what I'm referring to right now. But this is how they converge onto specific regions of the bulb, and this ability to recall a memory from a scent is typically because of its connection to areas like the amygdala and the hippocampus, which are strong regions in our brain that are related to memory. Now, gustation, again, which is our sense of taste. So taste receptors are located in taste buds on the tongue, in the papillae specifically, and all throughout the mouth. But This image shows how each taste bud contains about 50 to 150 taste receptor cells, along with support and basal cells. Now, each receptor cell responds to one of 5 basic tastes: sweet, sour, salty, bitter, and umami, which are the five types of taste. But signals from the taste receptor cells are carried by cranial nerves, 3 different cranial nerves, 7, 9, and 10, and they're all carried to the medulla, then to the thalamus, and finally to the gustatory cortex in the insula. Now, this taste, again, because it's not smell, smell is the only sense that does not have any thalamic relay, but gustation does enter through does access our brain through the thalamus. Now, taste works pretty closely with smell to create the full experience of flavor, because again, this targeted setup with the papillae on the tongue. So like, okay, the diagram, it shows how taste receptor cells are embedded in the papillae on the tongue and how their microvilli reach into the taste port to detect dissolved molecules, which is from what we're eating. It's a very targeted setup, so each receptor is sensitive to a specific chemical property, which enables our ability to have like precise tasting. Like, we know what we're eating when we're eating it. Again, taste and smell work together to create the full experience of flavor, which is why our nose is stuffed. Like when we have a cold, or when like something is blocking us from smelling to the full extent, we also have trouble tasting to the full extent. The integration of both the senses allow us to enjoy and evaluate kind of what we're eating and like whether or not we like it or not. Now, as for neurocommunication and signal transduction, this slide just kind of shows how chemical signals can be turned into electrical signals, or not really shows, but it tells you that in order for us to kind of process our senses, they need to switch from chemical to electrical signals in our brain. So in smells, odorants bind to G protein couple receptors, or GPCRs, which trigger a cascade that opens ion channels and creates action potentials. Taste transduction varies by the type of taste. So salty and sour taste typically use ion channels, while sweet, umami, and bitter taste use GPCRs, or G protein couple receptors. These complex pathways let our brain interpret combinations of chemical stimuli and as like distinct taste or distinct smells that we're able to recognize. The brain decodes this information through a process called population coding, which I learned from the textbook. It uses patterns of like activated neurons to distinguish between thousands of different chemical stimuli. Now, as for the integration in the brain, so here you can see how smell and taste converge in the brain. So olfactory input goes directly to the limbic system, which links it to our emotional systems and our memory, like we mentioned before, the amygdala and the hippocampus, anything that has to do with that. Whereas gustatory signals are interpreted in the gustatory cortex. Now, this image specifically labels the insular cortex, amygdala, and hippocampus, which emphasize how taste and smell enter emotional and cognitive like processing loops. This is kind of like a childlike projection of kind of what's happening, but basically the pizza is, once you put it on the tongue, your taste buds able to absorb the broken down molecules, and then it is integrated into the different cells and clusters. After that, we send signals to let them know that we like have the receptor for this taste, and then it's integrated into the brain, and then our brain is able to recognize that this is what we're doing. These two systems, again, taste and smell, they work together to create the perception of flavor, mood, and sometimes even behavior. But this multisensory integration explains why losing smell or taste impacts not just eating, but emotional well-being as well. And finally, I just wanted to add this slide in to talk about kind of real-world applications of chemoreception. I personally experienced the loss of taste and smell during the COVID-19 pandemic in 2021. I lost my smell and taste for several weeks. I was positive for COVID for only a few days, and then after, I still suffered from those side effects. At that point, food had no flavor. I couldn't enjoy meals or even like detect anything. Like, I couldn't smell spoiled milk and not be able to tell you that it was smelled or spoiled. But this just shows like that eventually our olfactory neurons are going to regenerate, because thankfully both smell and taste have returned over time. I'm perfectly okay, but it just reminded me how chemoreception connects us to not just our environment, but to memory, safety, joy. Anything we enjoy, eating wise or drinking wise, is because of this. It's not just about detecting chemicals, it's about experiencing life to the fullest. But that was all. Thank you so much for listening. I hope you now have a better appreciation for the biology and complexity of smell and taste. Um, yeah, these senses are like oftentimes they're overlooked because it's something we experience every day. But um, once you lose them, it's hard to get them back. But I'd be happy to take any questions from anybody. Please let me know, and thanks for listening.